Welcome to the final video in our series on driving the power of single-cell technologies forward into the clinic with ScaiVision. I'm Sarah Carl, Director of Data Science at Skylight. In the last video, I talked about how we enable clinical translatability through applying explainable AI methods to extract the most relevant features from trained models. Today, I want to put everything together and walk you through an end-to-end example of how we used ScaiVision to discover a diagnostic biomarker in endometriosis, simplify it to a minimal panel of genes, and prototype a clinically relevant assay. First, I'll present the dataset, which is actually a pretty complex and challenging one. We generated this by performing single-cell RNA-seq on endometrial biopsies from patients either with or without confirmed endometriosis, together with our clinical partners at the University Hospital in Bern, Switzerland. For this project, we performed quite an extensive descriptive analysis, as well as performing biomarker discovery. And one of our first observations was that we couldn't see any significant differences between endometriosis and control in terms of the frequencies of major cell types. So it was clear that there wasn't a major shift in cellular composition in endometriosis. To dig into the different sources of variation in this dataset, we performed PCA on suitable counts, and we immediately saw that the strongest variation was being driven by the menstrual cycle phase, which makes sense as it completely remodels the endometrial tissue. We also saw moderate technical effects stemming from both sequencing batch and interpatient heterogeneity. Endometriosis status showed only a weak correlation with some of the more minor PCs. All of this work is currently in our preprint on bioRxiv and hopefully will be published soon. After extensive pre-processing and data integration, we went ahead and trained ScaiVision models to predict endometriosis status. And we were successful in obtaining models that showed decent performance in a 5-fold cross-validation setup. So despite the weak underlying signal, our models were able to find it. As I presented in the previous video, we then applied integrated gradients to extract the most predictive features from the well-performing models. At first, we kept our signature fairly broad, comprising around 200 genes. As a first translational step, we compiled bulk RNA-seq data from the same type of endometrial biopsy samples, combining data that we generated ourselves as well as an independent cohort for a total of 104 patients. We used these data to build predictive models with the pre-selected signature genes as input features, and we were again able to achieve an AUC of 0.8 in predicting endometriosis. This step was important because it both validated our gene signature on a completely new patient cohort, as well as showing that we could recapitulate the single-cell signal in bulk data, even though when our collaborators performed classical differential expression analysis on these data, they didn't find any significantly differentially expressed genes between endo and control. After seeing the initial results in bulk RNA-seq, we decided to see if we could push it even farther along the translational path and move into qPCR. For this type of assay, we would ideally need a much smaller gene panel. So we applied an iterative strategy for feature selection in order to reduce the gene signature to the minimal set which retained good performance. This turned out to be 7 genes, which interestingly showed expression across a variety of different cell types in the single-cell dataset, including both endothelial and epithelial cells, as well as smooth muscle cells and some immune cell populations. We used this gene panel to design and prototype a clinical assay, starting from a sample collection kit, which we tested in the hospital, then moving to the actual qPCR assay, and then a software application to analyze the qPCR results and generate a report and a prediction. This assay has now been taken over by Hera Biotech, a women's health-focused biotech company in the US, with the aim to perform further clinical validation and eventually commercialize a diagnostic assay. Thanks for joining me throughout this video series. I hope that by providing more detail on the unique features of the ScaiVision single-cell analysis platform and showing some examples of its application, I've convinced you that with the right techniques, single-cell is ready to move from preclinical R&D into translational research and clinical trials. At Skylight, in addition to developing our analysis platform, we're also generating clinical-grade single-cell data from large patient cohorts for biomarker discovery. For example, we're currently kicking off a project in inflammatory bowel disease. We partner with pharma, biotech, and clinicians in order to address the toughest challenges in precision medicine. Please reach out if you're interested in getting access to our single-cell data, generating data from your own clinical or preclinical samples, or applying our analysis platform to your own existing data. Thanks for watching.